I can start to introduce Professor Hopi Hoekstra, another distinguished professor from Harvard, and Hopi got the PAG degree in the year 2000, and by the as a Harvard Hills product fellow, and in the year 2016, she was elected by the National Council member. So it's you can see it's tremendous. It's a very excellent work she done already, and now she was Harvard Hills Medical Institute about the principal investigator, and did a lot of good work and have a lot of award and also publication. So now she's a leading scientist in the genetic basis for the adaptation from morphology to the to the behavior. So I think it's maybe someday we can get the good news from Stephen Hall or maybe Sweden, and then so it's time to give the turn to the Hopi. So welcome.
Great, thank you so much. Can everybody hear me okay?
Yeah.
Okay, perfect, thank you. So let's see. First, I want to thank the organizers for the invitation. Like Keith, I'm disappointed not to be in San Diego, especially because here in Boston, we just had a snowstorm. Nonetheless, I'm really excited to tell you about the genomic basis of adaptation, a story from wild mice, a tale of tales. So we're going to really switch gears from topics that Keith discovered, and discussed. And although I'm trained as an evolutionary geneticist, today I'm going to push the boundaries of that and talk a little bit about organismal biology, evolution, behavior, and development, all in the context of genetics. So let's see. Before I begin, I just want to make sure to acknowledge the folks who did the work that I'm going to talk about today, in particular, Evan Kingsley, who started this project as a graduate student. He's now moved on and is a postdoc in Cliff Tabin's lab at Harvard Medical School. And he was quickly joined by Emily Hager, who has recently graduated and also stayed in the Boston area as a postdoc at BU. So throughout the talk, when I say we, what I really mean is Evan and/or Emily. So the major question that unites all of the work in my lab is this one: how do organisms adapt to novel environments? We think about this in two complementary contexts. First, sort of the classic context of, when organisms colonize new habitats. So a classic example of that is when, let's say, animals move from mainland habitat to a unique island habitat. But in recent years, we've become increasingly interested in another complementary scenario, that is when the organisms stay put, but the environments around them change. So whether that has to do with man-made change like agriculture, or climate change more generally. So to address this question, one of the approaches that we take as a geneticist is I first start with the question of trait variation, that is identifying traits that are one, variable within or between populations, and two, that we think are ecologically relevant. Then the hard work starts because we spend a lot of time trying to identify genomic regions, genes, and or mutations within those genes that give rise to variation in traits. Now, for many geneticists, this sort of represents the end game, but for us, it really represents a starting point, because once we have a gene in hand, we can start to ask questions about mechanism. In other words, we can start to better understand the evolutionary processes responsible for that trait variation, as well as the molecular mechanisms by which changes in a particular gene act through development to produce the variation in the trait of interest. In other words, we want to know both why traits vary and how traits vary, and to do that, we start at the level of genes. So over the last several decades, our lab has contributed to a growing number of examples in which we can identify genes, understand how they act through development to change morphology, and ultimately how that morphology affects fitness. So our lab started by looking at cryptic coloration, whether that's variation in pigment or patterning, and identified genes and even mutations that we were able to link to fitness differences in the wild. Sort of bolstered by this success, we more recently asked ourselves if we could use this same approach, not just to genetically dissect morphological variation, but could we apply the same approach to behavioral variation? Now, while this comes with a whole suite of additional complications, from the ability to measure complex behaviors to dealing with what is arguably the most complex and heterogeneous organ in our body, the brain, nonetheless, we've been able to make some progress in identifying genes that, for example, in this case, contribute to differences in parental care by comparing promiscuous and monogamous species of mice. But as we thought about that question that I raised at the beginning of the talk, that is, how do organisms adapt to their environment? Often it's not morphology alone that changes or behavior alone that changes, but they change in concert and in some cases even co-evolve. So this is the framework in which I want to present data to you today, that is genetically dissecting morphology and understanding how morphology can affect behavior. So to start, let me introduce you to the main player of today's talk. This is the deer mouse, otherwise known as Peromyscus maniculatus. It's the most widespread and abundant mammal in North America, and it's found in a wide distribution of habitat types across its range. It's been the subject of many ecological studies, and in the early 1900s, over a century ago, a complete taxonomic description was published by Wilfred Osgood, who drove around North America and characterized variation within and between populations within this single species. And one of the things he noted was that this species essentially comes in two different types, either a forest form or a prairie form. In other words, he claimed if you caught a mouse and showed it to him, just by looking at it, he could tell you if it came from the forest and the prairie or the prairie. And that's because these mice differ in a suite of phenotypic traits. Forest mice tended to have darker pigment, longer whiskers, bigger ears, larger feet, longer tails, and also preferred to climb and seemed to climb better than their prairie counterparts. What's even more remarkable is this whole suite of traits likely evolved just in the last 10,000 years. So as the glaciers retreated northward, this ancestral prairie type could colonize this new forested habitat and adapt to life in the trees. Even more recently, using molecular tools, we were able to show that this sort of evolutionary experiment has run twice independently on the West Coast and the East Coast of North America. So as an evolutionary geneticist, this was quite exciting, because what it means is we have two natural evolutionary replicates, the evolution of a forest to a prairie form that occurred independently on both coasts. So this led us from our molecular lab out into the field to catch mice. In the West Coast, we traveled to Oregon and captured mice from the coastal forests of Oregon and drove inland to the prairie habitat in Central Oregon. On the East Coast, we already had mice from southern Michigan from a prairie habitat, and we complemented that with mice we collected in the eastern forests in Pennsylvania. So now we could bring these mice back into the lab where we could study them under controlled conditions and look at their traits. So for the rest of the talk, I'm going to focus on these four populations, and I'm going to color code data from the forest in green and the prairie in brown. And the mice from the West Coast are going to be in slightly darker hues than the East Coast. So of all the traits that we looked at, today I'm going to focus primarily on the tail. And we were interested in the tail for two reasons. First, because the tail is highly variable. So here what I'm showing you is the tail to body ratio on the vertical axis across a number of populations or subspecies. And what you can see is that there's a large range in tails within the single species. Even more interestingly is that this variation in tail length correlates with habitat. That is, mice with the shortest tails all came from prairie habitat. Mice with the longest tails all came from forested habitat. And those that came from either intermediate or mixed habitat had intermediate length tails. So once in the lab, we could ask ourselves, do the mice that we captured in the field actually have differences in tail length as Osgood predicted over a century ago? And what we found is indeed they do, both in the field where we captured them and also that's maintained across generations in the lab. So for example, you can see here, we're looking again at tail to body length ratio. Forest mice have tails that are longer than the length of the body, whereas prairie mice always have tails that are shorter than the length of the body. There's two other patterns I want you to note. First, the difference between forest and prairie mice within a location, for example, on the West Coast, the magnitude of difference is approximately the same on the West Coast and the East Coast. But the second thing you may note is that within a habitat type, West Coast mice tend to have longer tails than East Coast mice. And we're not exactly sure why this is, other than to point out that the forests on the West Coast aren't the same as forests on the East Coast, and similarly with the prairies. So because this trait is maintained across generation in controlled laboratory conditions, it suggests that there's a strong genetic component, which was exciting to us. But before we jumped into the genetics, one of the first questions we wanted to explore was why these mice may have different tail lengths. Well, Osgood postulated, and you may be thinking the same thing, that having a longer tail might in fact be beneficial for mice who climb in this new forested environment. This hypothesis was first tested by Elizabeth Horner when she was a PhD student teaching at Smith College. And she did what I would argue is a beautifully elegant experiment, although slightly gruesome. Okay, so here's what she did. She brought mice from the same species into the lab, a forest mouse shown in green and a prairie mouse in brown. And she had them run across a narrow rod that connected two platforms that were elevated above the ground. And then she counted the number of times they made an error, in other words, fell off the rod. And what you can see, maybe as you would expect, is that prairie mice had a higher number of errors or lower performance compared to forest mice. And here's where the experiment gets a little bit gruesome. The next thing she did was amputated the tails. She let the mice recover for a few weeks and then reran them on this assay. Now, the more times the mice run on this assay, the better they get at crossing the rod. And so we would expect that their performance would improve. In other words, the dots would be lower. But instead, what she found was that either there was no difference in performance, or in fact, the performance was worse when the tails were amputated. So this tells us two things. First, it tells us that tails matter for climbing, most likely for balance. And second, that if tails matter more for forest mice, in other words, they rely on their tails more than prairie mice. So inspired by this experiment, we wanted to return to the same sort of assay, but improve it in two ways. First, we were interested not just in whether a mouse had a tail or didn't have a tail, but instead wanted to test these tail length differences that we observed in nature between a forest and a prairie mouse. And we wanted to quantify their performance in a much more automated and quantitative way, not just how many times they fell off the rod. Now, while I appreciate this doesn't this simple behavioral assay doesn't capture all the complexities of climbing in a real life forest, we think it does capture ecologically relevant differences because these mice actually cross narrow rods in the field. So next, what I want to do is show you two videos that just give you a sense for the differences in climbing performance. And importantly, these represent the median behavior of each of these two ecotypes. So these are not extremes. So first, I'm going to show you the performance of a prairie mouse. As you can see, it's in this behavioral assay, two platforms connected by the narrow rod. And I want you to pay attention to two things, not only its overall performance, but how it uses its tail, both as a stiff rod for counterbalance, but also it wraps its tail around the rod to prevent falls. So here the mouse goes, and this Eastern Prairie mouse, which is naive, it's been introduced into this behavioral assay for the first time. And we videotaped it for 5 minutes. You can see that it nicely crosses the rod. And then, as I mentioned, we can automate the analysis of this behavior. This is in stark contrast to the Eastern Prairie mouse, which you can see here. It gets on the rod, immediately falls off, tries to right itself, and frantically attempts to get back to the platform. And again, we can automate, in this case, the poor performance of a prairie mouse. So using this assay, we can start to quantify differences in behavior between the two, or performance of the two. So as the videos suggest, we see a big difference in performance between forest and prairie mice. So if we consider, for example, the fraction of time spent upright, forest mice spend approximately 85 to 95% of their time upright, whereas the poor prairie mice spend the vast majority of their time upside down. This is for 2 reasons. One is, when a mouse is upright, a forest mouse will spend more time upright before falling compared to a prairie mouse. And second, if a mouse does fall upside down, a forest mouse will right itself more quickly than a prairie mouse. Moreover, once upright, forest mice will run across the rod more quickly compared to prairie mice. So these data together suggest an evolutionary explanation for why tails are longer in forest mice, is that it helps with balance and climbing in their forested habitats. But importantly, you have to remember in this particular assay is that forest mice not only have longer tails, they have larger feet, longer whiskers, and other traits, which confound this correlation. So I'm going to return to the question of the direct relationship between tail length and performance at the end of the talk. But for now, I want to switch to another question, and that is, how do mice get longer tails? So to address this question, we're going to focus on 3 levels: morphology, genetics, and development. And I'm going to start with morphology. So if we think about a mouse's tail, it's comprised of caudal vertebrae. And in the simplest way, we can think of a tail getting longer using 2 strategies. One, you can keep the number of caudal vertebrae the same and simply increase the length of each vertebra, resulting in a longer tail. Alternatively, you can keep the length the same and just add caudal vertebrae to the end of the tail. So to ask which strategy or combination of strategies evolution has used on the west and east coast lineages, what we did is we took X-rays of mice and automated the analysis of 3 traits. One is shown in black, overall tail length. In orange, we measured the length of the longest vertebra, in part because vertebra length is correlated along the tail. So we focused on just one vertebra. And then we counted the number of vertebrae. From these data, what we found is that forest mice on both coasts have both longer and more vertebra. So for a given body size, using sacrum length as a proxy, you can see that forest mice shown in green always have longer vertebrae than prairie mice shown in brown. Moreover, they also have more vertebra. So for example, on the West Coast, an average forest mouse has 28 or 29 vertebra, where its prairie counterpart has 24 or 25. Next, we wanted to know, of these 2 strategies, how does each contribute to the difference in overall length of the tail? So here what you're looking at is on the horizontal axis, the marching down the length of the tail, and we're looking at the cumulative tail length on the vertical axis. So the question is, how does a short-tailed prairie mouse get to the length of a long-tailed forest mouse? And it turns out that adding vertebrae gets you 50% of the way there, and elongating vertebrae gets you the other 50%. In other words, the 2 strategies contribute equally to the difference in overall tail length. And remarkably, this is true not only for the West Coast, but also for the East Coast. So the question then becomes, in these independent evolutionary replicates, why is there such remarkable convergence in this phenotype? Well, we came up with 3 hypotheses. The first one is it could be genetic constraint or pleiotropy. That is, mutations that are selected because they increase the overall tail length affect both vertebrae length and number within a given mutation. Second, it could be a functional explanation. That is, that there's some optimal kind of trade-off between vertebra number and length that allows the mouse to keep its tail stiff to use as a counterbalance, but also maintains enough flexibility that it could wrap its tail around the rod. And third, it could be chance. In other words, there could be lots of mutations that contribute to overall tail length differences, and roughly half of them either arising as new mutations or as pre-existing mutations in the ancestral population. Roughly half of them affect vertebra length and the other half vertebra number. And what really matters is just overall tail length, not how you get there. So to test the hypotheses, we started with this first one, that is looking at genetic constraint, because we could easily test this using genetic crosses. So what we did was we independently on the east and west coast is we took short-tailed prairie mice that conveniently have tan-colored chromosomes, crossed them with long-tailed forest mice to make F1 hybrids, which of course have a chromosome from each of the parents and notably have an intermediate length tail. And then intercross these F1 hybrids to make this genetically heterogeneous F2 population in which each individual has a different combination of alleles from the forest and prairie parent, and hence have a wide range in tail lengths, and also we're able in this population to dissociate tail length from other ecotype specific traits. Then in these F2 populations, we could ask with their shuffled genomes, is there a correlation between vertebra length and number? What we found in both the West Coast and the East Coast is there's no correlation between vertebra length and number. In other words, there are at least two independent genetic mechanisms that affect tail length. In other words, there's the mutations affect independently vertebra length and vertebra number. So we could cross off this hypothesis that it's genetic constraint or pleiotropy. And next we turn to this functional explanation. So using a biomechanical approach by carefully analyzing videos of mice crossing rods, we were able to show that there doesn't seem to be an evolutionary trade-off between vertebrae length and number, or stiffness and flexibility in the tail, at least in this particular assay. So I'm not going to tell you about the details, but this work is recently published, and I encourage you to have a look if you're interested. So this left us with this final explanation that potentially this convergence in morphology is just due to chance. But to test that, what we really wanted to know was something more about the alleles that contribute to overall tail length difference. So this leads me to the next question of how do mice get longer tails from a genetic perspective? So as I just showed you, we did these two large crosses independently on the east and west coast. We made approximately 500 F2 hybrids. We measured their tail traits. And next what we did is we genotyped them at hundreds and thousands of markers across the genome and asked, is there a genetic correlation between their genotype at any marker in the genome and their tail phenotype? In other words, we did QTL mapping. So first I'm going to show you data just from the cross on the East Coast. When we considered overall tail length, what we found was there are 6 chromosomes shown here that show a statistical correlation between genotype and total tail length phenotype as measured through a LOD score. And the statistically defined regions are highlighted here by these gray blocks. So in other words, it doesn't look like the tail length difference is controlled by a single gene, but on the other hand, it's not controlled by hundreds of genes. And this makes sense in that we think that this difference in tail length evolved over just a few thousand years, making it difficult to imagine that hundreds of genes could accumulate between the two ecotypes. But not only did we measure total tail length, we measured its component parts. And we can ask now what QTL, or what regions of the genome, correlate with the length of the longest vertebra. And what we find of the 6 QTL associated with total tail length, 3 are associated with longest vertebra. And in a remarkably symmetrical result, the other 3 QTL associate with the number of vertebrae. So this is interesting for 3 reasons. First, it explains nicely why we were able to dissociate length of vertebra from number of vertebra in those F2 hybrids, because the causal mutations are on independently segregating chromosomes. Second thing of interest is that the QTL associated with longest vertebra explain roughly the same amount of variation in tail length as the QTL for number of vertebrae, about 20% each. And then finally, in all 6 of these QTL, it's always the forest allele that's associated with longer tail. This is significantly different from random, suggesting the action of natural selection in driving tail length differences. So these are the results on the genetic architecture associated with tail length differences on the East Coast, but what's happening on the West Coast mice? We did the same experiment, and like on the East Coast, we found a handful of QTL, which are highlighted here in pink. But what we really wanted to know is, do any of those genomic regions overlap between the 2 lineages? And what we found, quite to our surprise, despite the fact that the morphology was almost identical, that there's almost no overlap, with the exception perhaps on chromosome 20, where the 2 regions, statistically defined regions, abut each other. There's absolutely no overlap in the mutations that are associated with differences in tail length on the East and the West Coast. So one of the things you may notice when you look more carefully at the West Coast is that there's one QTL in particular that may catch your eye. So I just want to spend a moment talking about this QTL found on chromosome 15. It's remarkable both because it shows a strong association with tail length differences, but also the shape of this particular peak shows that it's flat. In other words, the association remains high across approximately half of the chromosome. So this caught our attention, and we decided to investigate further. And what we found was that this part of the chromosome had a very interesting pattern, in that the left half of chromosome 15 showed no evidence for recombination in our F2 hybrids. When we looked at variation in natural populations, we saw there was an increase in average divergence, or as measured by FST, on half of the chromosome relative to the other half. And finally, there's a high level of linkage disequilibrium on the left side of the chromosome relative to the right. Well, it turns out, as we confirmed by long-read sequencing, that this represents a large, simple inversion in which there's a derived inversion is associated with longer tails. This inversion is about 40 megabytes in length and represents about half of the chromosome. Interestingly, as the QTL data suggests, the inversion genotype correlates nicely with tail length and in the direction we expect. That is, if you have 2 forest alleles at the inversion, you have a longer tail than if you have 2 prairie alleles. And if you're heterozygous, you have an intermediate phenotype. This inversion explains about 12% of the variation in overall tail length, representing about almost 3 millimeters length difference. The inversion is not only associated with tail length, but its frequency is associated with habitat. So if you collect mice across a cline going from the forest populations to the prairie populations, what you find is that tail length is high in the forest. And then as you transition to prairie habitat, you can see tail length decreases. This cline in tail length is perfectly mirrored in the allele frequency of this chromosome 15 inversion and differs significantly from the genome-wide patterns. This again implicates a role for natural selection. And then finally, if we look at the frequency or distribution of this inversion across the range of this particular species, what we find is another interesting pattern. So each dot represents a different population that we sampled. The size of the dot represents the number of individuals in that sample. If that sample comes from a forest population, the circumference is in green, and a prairie, it's in brown. And the pie charts represent the frequency of the inversion, the derived inversion, shown in green. So the 2 things to note are, first, that the inversion is found only on the west coast of North America and is completely absent from the East Coast, even in long-tailed forested mice. And second, in the West Coast, The inversion is at much higher frequency in forested populations than in prairie populations. So that's nicely captured in this graph shown on the right, that you have long-tailed mice on both the west and east coast of North America relative to prairie mice, but the inversion is only found on the west coast. So in other words, together, this inversion is a nice example of how this remarkable convergence in morphology is not due to the same genes. In this case, the inversion is found on the west coast, but not on the east coast. And if you're interested in this inversion, we have a bioRxiv preprint up that I encourage you to have a look at. But for the rest of the talk, I'm going to focus on the East Coast cross, which I showed you initially. And in particular, I'm going to focus on one genomic region, and it's one that's associated with the number of vertebrae, in part because this is a trait that's established during embryogenesis. So the number of vertebrae is determined before birth. And in particular, I'm going to focus on this QTL on chromosome 8 because it's the strongest and the most consistent QTL in our data set. So as I mentioned, vertebra number is established at birth. So you can see when mice are born, forest mice have more vertebrae than prairie mice. So that suggests that there's something going on during embryogenesis that gives rise to this difference. So we decided to take an RNA-seq approach and pick two critical points during tail development, dissected out the tail buds of forest and prairie mice and compared gene expression. Perhaps not surprisingly, we found that thousands of genes are differentially expressed, even though these mice are separated by just a few thousand years. So this approach alone wasn't going to narrow in on any particular candidate genes, but we could take this RNA-seq data and intersect it with our QTL data to ask, of the 140 or so protein coding genes found within this genomic region, how many of those show differential expression of the thousands that are differentially expressed? And what we found is 25 candidate genes. But 25 candidate genes is still a lot to functionally interrogate. So we took advantage of the fact that there are many genes that have been identified in laboratory populations of mus musculus, presumably spontaneous mutations that occurred in laboratory colonies, and intersected this data set of 155 genes and found 3 of them that are differentially expressed and in our QTL. So what are these 3 genes? Well, perhaps not surprising, they're all important developmental genes. 2 of them are Hox genes, and one is SP5, a gene important for early embryonic patterning and specifically involved in Wnt signaling. Of these 3 genes, one in particular caught our attention, and that is HOXD13. So if you remember back to your developmental genetics classes, one of the things that's so remarkable about Hox genes is they're collinear. In other words, the way that they're organized along chromosome mirrors where they're expressed in a developing embryo. So HOX13 genes are at the end of this genomic cluster and are expressed at the very tail end of a developing mouse embryo. So it's expressed in a relevant place. Moreover, beautiful developmental studies led by Denis Deboul and the Deschamps lab have shown that when you turn on HOX13 paralogs in mouse development, it causes earlier axial termination. In other words, you get a shorter tail. When we look at our gene expression patterns, what you can see is indeed the short tail mice have high HOXD13 expression early on in development, and our long-tailed mice only see this increase in HOXD13 later on, consistent with patterns found in mus. Moreover, when we look at HOXD13 allele-specific expression in F1 hybrids, we find that these differences in gene expression are largely recapitulated at the allele level, implicating a role for cis-regulatory mutations driving this difference in HOXD13 expression, and consistent with HOXD13 being in our QTL peak. So this gene has become our favorite candidate gene, and we've started to interrogate the underlying molecular mechanisms. The first thing we did was compared the protein coding region between forest and prairie mice. And here I'm showing you a region of HOXD13 and comparing deer mice, shown on the bottom, to a number of other mammals. What you can see is that the gene is largely conserved across mammals, and in fact is identical between forest and prairie mice, except for one site shown here in blue, in which prairie mice have an insertion of the T that's not found in deer mice, and in fact not found in any other mammal. This is relevant because this is in a string of alanines, and we know that changes in the size of this repeat region can have skeletal effects in humans. So to test the role of this particular mutation, we engineered in laboratory mice this mutation and asked, does it cause a difference in number of tail vertebrae? And in fact, what we found was there was no measurable effect on tail vertebrae. So this then led us to focus on cis-regulatory changes, which is initially how we found this as a candidate gene anyway. So to try to identify mutations in or around HOXD13, we did comparative attack-seq, looked for different regions that were differentially accessible, open chromatin, and identified several candidate regions shown here in red, have cloned out those regions and are now running luciferase assays in a number of different cell types to see if they drive differential reporter expression between the two. So I don't have those data yet. You'll have to stay tuned to see if we can implicate particular regions and even mutations in those regions driving differential expression. So in addition to sort of figuring out the molecular mechanism, we wanted to understand also how HOXD13 might play a role in development. Now, recognizing that this is a more genomics audience, I wanted to just spend 3 quick slides summarizing years worth of work to tell you how we think HOXD13 acts through development. So again, this number of vertebrae is established during embryogenesis, and we know from laboratory studies in mouse that there's this key window in which tails elongate. Through, again, a number of beautiful studies from developmental biology, the quick explanation for how tail vertebrae develop is that they develop from somites, and somites are derived from a material at the very end of a developing embryo called the pre-Semitic mesoderm. I like to think of the pre-Semitic mesoderm as a loaf of bread, and that loaf of bread is sliced into somites, and those somites then develop into vertebrae. So with that background in mind, we came up with 2 hypotheses about how a forest mouse, shown in green, can have more somites and more vertebrae. The first hypothesis is that they have the same pre-Semitic mesoderm. The loaf of bread is the same size, but you just slice it more quickly. The slices are thinner, and that's how you get more vertebrae. Alternatively, you could start off with different sizes of pre-Semitic mesoderm or loaves of bread, but the rate at which you slice remains the same. This could also result in more vertebrae. So to test these 2 hypotheses, we did a number of experiments. I'm just going to show you some key results. First is we found no difference in the somite size, which would be predicted if we made more slices that were each thinner. So if we look across developmental time on the horizontal axis, you can see the length of the last form of somite didn't differ between the 2 ecotypes. But by contrast, what we found was there is a difference in the pre-Semitic mesoderm length and in the direction we expected, with the forest mice having a larger pre-Semitic mesoderm. So in other words, we didn't find any evidence supporting hypothesis 1, but instead it looks like forest mice get longer tails, at least in part because they have a larger pre-Semitic mesoderm. So how does having a larger pre-Semitic mesoderm link to the data I showed you earlier, a role for HOXD13? Well, again, we know from developmental biology that the pre-Semitic mesoderm derives from what are called neuromesodermal progenitor cells, or NMPs. When we looked at NMPs by doubly labeling cells in the contraneural hinge, what we saw is that there are in fact more of these progenitor cells in forest mice compared to prairie mice. This suggests that the reason that forest mice have larger pre-Semitic mesoderms is because they have more progenitor cells. And excitingly, we know from studies in laboratory mice that HOXD13 likely plays an important role in the proliferation and maintenance of these NMP cells, thereby providing a more direct link between D13 and expression differences, this difference in pre-Semitic mesoderm and ultimately in number of vertebrae. So now what I'd like to do is just step back and summarize what I've told you. So the study started with a comparison between forest and prairie mice, a natural history observation made over a century ago. I showed you that these forest and prairie mice, when we brought them into the lab, showed pretty dramatic differences in climbing behavior and performance, with forest mice performing better in this particular climbing assay. From a morphological perspective, I showed you that forest mice have longer tails, that this is maintained in the lab, has a strong genetic component. And they have longer tails both because vertebrae are longer and they also have more vertebrae. From a genetic perspective, this is likely due to, at least in part due to a difference in Hox D13 expression. And from a developmental perspective, Hox D13 likely affects the pre-Semitic mesoderm size, which gives rise to the differences in number of vertebrae. Now, as you may note, there's still one link that's missing that I hinted at earlier in the talk. That is, I haven't yet shown you that tail morphology has a direct effect on climbing behavior, only that the two are correlated. So the last piece of data I want to show you makes this direct link between differences in tail morphology and climbing behavior. So again, we could return to this wonderfully rich resource that is these large F2 recombinant hybrid populations. Of these populations, I already told you, we measured their tail morphology and its component parts, but we also now ran each of them through our climbing assay and could ask, in these mice that have different length tails and different component parts, is there a correlation between tail morphology and climbing behavior when we could control for all of those other traits like foot size, whisker length, etc. And what we found is, indeed, there's a strong correlation between morphology and performance. And importantly, when we looked at, in this canonical correlation analysis, and when we looked at what traits or variables loaded onto these axes, what we find is that small mice with large feet and most importantly, long tails perform better. That is, they spend more time upright, and when upright, move across the rod at a higher speed and spend less time upside down. So in other words, this provides a direct link between differences in tail length and climbing performance. So what I've hoped to show you today is that using this approach, even in non-model systems, we're able to identify strong candidate genes, provide mechanisms by which those genes act through development to produce variation in morphology, and how variation in morphology can affect behavior. And ultimately, we hope to show how it affects fitness. Current work in the lab is really focused on the right side of this diagram, in the sense we're now looking for genes that act in or on neural circuits that affect climbing behavior independent of morphology, and ultimately to understand how that affects fitness. So with that, let me thank again, Evan and Emily, who really led the work. They worked in strong collaboration with Olivia Herringmeyer and Brock Wooldridge, two graduate students in the lab. They were helped by this team of technicians, a great team of undergraduates who have helped with this project. And Xiaonan Hu and Kelsey are both postdocs in the lab who are working on this project, whose work I didn't have time to tell you about today, and some collaborative work with Adam Hantman. This work was supported by HHMI, Harvard, and the Museum of Comparative Zoology. And with that, I'm happy to answer any questions. Thank you very much for your attention.
Thank you, excellent talk. So now we take a question. In the web, I have 6 questions already.
Okay.
Yeah.
Okay, should I just take those as Keith did?
Yeah.
Okay. So is another explanation of the finding that both number of vertebrae and length of vertebrae are under selection, i.e. natural selection on tail length increase both the number of vertebrae and the length? Okay, so what I can tell you is, if I am understanding this question right, I presented 3 hypotheses. One, that a that it's pleiotropy, which I think we can clearly rule out. We didn't find evidence for a functional explanation, that is, that it's that having a tail that, if you can imagine, if you just elongated vertebrae, your tail would be really stiff, but it wouldn't be flexible. If you just added vertebrae, you'd be really flexible, but it wouldn't be stiff. And instead, what we found was it didn't matter how you got a longer tail, but what really mattered is that your tail was longer. And that matters because of the mass of the tail. And the larger the mass of the tail, the more effectively the tail can be used to help with your balance. So we think that's the leading hypothesis for the trade-off between the two. And from a genetic perspective, what we think is that there are equal numbers of mutations either that are possible or that are already existing, segregating in the ancestral population that affect number and length of vertebrae, and they're just being selected sort of at random in both populations. But to really test that hypothesis, what we need to do is find those mutations and see if they're segregating in the ancestral population. And that's why, in part, why we're spending so much time trying to dissect the genetic architecture of tail length. Let's see, next question. Do you see inversions in chromosome 15 for the Midwest mice? Knowing the West Coast mice have an inversion in the East Coast don't. Okay, that's a great question. And so in fact, what we did to address that question in part, as well as other questions, is we went back and sampled mice from over 300 mice from approximately 40 populations covering the whole range of North America up through Canada down to Mexico, and are looking at the frequency of the inversion. What we find is that it is really restricted to the West Coast. We don't find it so much in Central North America, and we never find it in North America. But this biogeographic break, that is this sort of having the West Coast and the East Coast being separate, is actually a pattern found in a wide range of vertebrates. And so there's some sort of biogeographical history that explains that. But we do find it in several interesting populations in the Southwest US that I didn't have time to talk about today. Okay, so the next question is about what do we know about the vertebrae length? Well, so today I really focused on the dissecting vertebrae number. Vertebrae length is a little bit more tricky, but one of the things I didn't mention is that this inversion that we found on the West Coast doesn't affect vertebra number, it affects vertebra length. And so in that sense, we're starting to really look at vertebra length in the West Coast populations more carefully. But of course, we're also interested in that on the East Coast. We haven't focused so much in part because when we first did this analysis and Hox genes popped up associated with vertebra number, we focused on that first. So give us some time, and we're really interested in the underlying genetics of vertebra length. But we haven't made much progress on the precise molecular mechanisms yet. Okay, let me do this question next. So it's shorter. And that is, what happens if you cross West and East Coast mice? I love this idea, and it's something we thought about very early on when we first started this project, but we focused on the crosses within a lineage initially. We just, before the holiday break, set up the crosses between long-tailed mice from the East and West Coast and short-tailed mice from the East and West Coast, and the reciprocal crosses, in some sense trying to see if we could do sort of a genetic complementation test. And what we would predict based on what we've learned so far is that we could get mice that were super long-tailed, and we could get mice that were super short-tailed, and everything in between. So I love that experiment, and stay tuned because it's underway. Okay, here's another great question about are shorter tails, why do prairie mice have shorter tails? Is it for decreased predation in prairie environments? Yep, so I sort of presented this story as long tails are important for forests, but didn't really talk about the reciprocal side. And that is, we think that prairie mice have short tails, and short tails are selected for in prairie environments. In other words, there's divergent selection working in opposite directions in those two environments. In terms of explanations for why they have shorter tails, there's a few, although we haven't tested any of these. And as you can imagine, it's rather difficult to test. One, it could be that having shorter tails reduces predation. Tails also are a part of the body that can often get injured, so it reduces injury risk. And also tails are developmentally expensive to produce. So if you don't need a long tail, why bother producing one at all? So we think that's why prairie mice have short tails, but the benefit in terms of climbing performance and potentially other factors outweigh those costs in forest mice that are living up in the trees. Should I do one more?
Yeah, we have time.
Okay, this is a great one.
3 minutes. Yeah.
Okay. Perfect. And then maybe I'll get to the longer question above. So this question was, have you checked the correlation between performance and each component of tail length? Right. So the data that I showed you at the very end is simply looking at overall tail length and climbing performance. So we are analyzing that data right now. So what happens to a particular mouse if it has a long tail, but it's long just because it increased vertebra length, but not added number? And alternatively, the other extreme, what if you have a long-tailed mouse that has lots of vertebrae, but each one is small? I think that's such an exciting question. And one of the reasons we did that experiment, but we're in the midst of analyzing that data right now. And stay tuned. We have the figures are being drawn up as we speak. Okay.
Hopi, this is Darren. I just wanted to say, if you're comfortable with going through the questions, I see there's a total of 15 and a few left. If you have the time to answer all of them, that would be great. And then, we can end in a few minutes.
Okay. So should I end there and then I'll?
No, you can keep going.
Okay. Tell me when to stop because I could talk about this all day long, right?
Okay. Well, once you finish the last question, so I think we're almost there.
Okay. Okay. This is a question about the attack-seek data. Have we looked at what transcription factor motifs are in that region and whether TFs and other loci associated with the number of vertebrae? That's a great question. We did indeed look at transcription factor binding sites within those regions that look differentially accessible between forest and prairie mice during tail elongation during embryogenesis. There are transcription factor binding sites. There wasn't anything that popped out as like a smoking gun obvious site in which the transcription factor had previously been implicated in tail length differences, but there were a number that looked promising. So what we've decided to do is wait for the luciferase assays to get run, and that's a little bit tricky, as I sort of implicated, in the sense that in order to get results that are biologically meaningful, we have to run them in appropriate cell lines, cell types. And so we're trying a number of different cell types ranging from sort of hex cells to more sort of appropriate tissue related cell lines. And then I think if those give us promising results, we'll go back and look at the transcription factor binding sites within those regions because there was, again, as I mentioned, no smoking gun. Did we try to check gene co-expression, which could probably tell you the key genes that regulate tail differences? So we have started to look at more genetic network analyses types approaches, especially in those RNA-seq data. I don't have data from those yet, but that's definitely an approach that we're hoping can provide some insight into at the very least, what pathways may be different, not just those associated with HOXD13, but remember there are these other, in the East Coast, at least 5 different QTL in which we haven't implicated yet genes or developmental mechanisms. So I think that could be really helpful. I think that's a great suggestion that we're considering. I'm going to keep going until somebody tells me to stop.
Yeah.
One more?
One more question.
Okay.
Yeah.
Have you tried a GWAS strategy to identify the candidate loci? So this is a really good question. And so I sort of hinted at this data. On the West Coast, we have long and short-tailed mice. And across Oregon, you go from forest and then you quickly transition to prairie habitat. And in the middle of that transition zone, you have mice with a wide range of tail lengths. And that to us struck us as a potentially exciting population and to do GWAS studies within that population. So we've resequenced mice from that population. And it turns out that we see some associations, but what we really need to do is go back and resample because we have something like 50 mice. And to do this really well and to see those associations, we're going to need to have more mice and we're going to need to do a better job at our resequencing. But I think that's a very exciting approach to try to narrow in, to complement the QTL analysis and ask within those QTL, are there particular regions that we can identify through GWAS that will give us finer scale resolution, whether that's at the gene level or mutation level. So I think that's really exciting. We've thought about that and haven't yet fully done that. As you're getting, I hope, the impression, there's lots of exciting things to do. And one of the hardest things about this project is trying to prioritize and focus on just a few things because there's so many exciting avenues.
Okay, thank you. The two excellent speaker. And if you have question, you can email to Hopi or maybe Case. So I think we can stop here and maybe see you next year or maybe this year someday. Okay, thank you, Arvin.